Good morning, friends. Welcome to today's class. Let's start today's class with a quote. The journey of 1,000 miles must begin with a single step. We will be discussing the definition, basis, need, type, and related terminologies of the unit reproduction, as we have studied in the previous classes, the part of a typical flower with a reference to the essential and non-essential worlds. We will be discussing the main events of the reproduction by discussing the stages, that is pre-reproductive stages, the gamete transfer and fertilization, then the post-fertilization event in detail. Okay, we will be discussing the gametogenesis in plants, that is microsporogenesis, the formation of pollen grain, followed by the megasporogenesis, the formation of embryo sac. Then coming back to the gamete transfer, that is pollination types and agencies and their peculiar features of the flowers adopted. Then the fertilization process of syngamy and triple fusion, which happens in the typical embryo sac, and the post-fertilization events where the ovary is becoming fruit and ovule into seed, and also typical structure of a seed during germination. So these events will be discussed in detail. Today we will be discussing the pre-reproductive events in angiosperms, the gametogenesis of microsporogenesis and megasporogenesis. You may get a diagram-based questions for labeling in the board exams. So here it goes. In the first part, we will be discussing the microsporogenesis, that is the formation of pollen grains. The site of production is sporogenous tissues or special cells called MMCs, microspore mother cells. You can see the schematic diagram of an anther TS and showing the connective. You may get a diagram based question for labeling 1, 2, 3, 4, 5, and the histological perspective of various layered arrangement showing the different parts of epidermis, endothecium, middle layers, tapetum, MMC. Coming to the next part of megasporogenesis, you may get a diagram based question for labeling showing the embryo sac, and it will be formed from the nucellus tissue from a single MMC, megaspore mother cell, and the process is called monosporic development, that is formation of megaspore. Okay, drawing and labeling of the various parts and the functions, starting from the stalk, micropyle, the opening, and the filiform apparatus, finger-like projections and the synergids, the egg cells in the center, and the central cell with the central nuclei, and antipodals on the top with the callosal end. Okay, so we will be discussing it in drawing and labeling and identifying the parts and functions of gametogenesis, the pre-reproductive events. Okay. Hello. Good morning, friends. Welcome to today's class. Let's start the day's class with a quote. Purity, patience, and perseverance are the three essentials for success. Above all, love. Today we will be understanding the concept of megasporogenesis. Having studied the microsporogenesis, we will be doing the structure of a pollen grain, identifying the pollen grain from a bloomed up flower, and understanding the parts of a anther, then the pollen grain with exine and intine with the sporopollenin and the cellulospectin covers. Coming to the other aspects of labeling, and we will be understanding the parts of the typical gynoecium and the embryo sac of a typical anatropous ovule. Okay. Good morning, friends. Welcome to today's class. Let's start the day's class with a quote. As long as I live, so long do I learn. Today we will be discussing some important events in the life of a bisexual flower, the pollination, double fertilization and post-fertilization events in angiosperms. In the last class we have studied about the pollen grain structure. Today we will be discussing about the pollination. The transfer of gametes is called pollination in plants, where there are two types of self-pollination and cross-pollination. You can see the diagram and you may get the diagram based questions labeled as 1, 2 here in the diagram, and you are supposed to explain the self-pollination and cross-pollination. If the pollen grains are transferred from the anther of a flower to the stigma of the same flower or the stigma of the same plant flower is known as self-pollination. When it is happening to the other flower of another plant, it is called cross-pollination. We have defined it and you have written the definition also by seeing the diagram. Okay. After pollination, the next step happening is double fertilization, which is known as syngamy and triple fusion. You may get a diagram based questions to label the stigma, style or pollen tube and the embryo sac and the egg here in the bottom part of the egg apparatus as shown in the diagram. So why it is called a double fertilization? Because each pollen grain will start germinating to release two gametes, two male gametes. One of the male gametes will be fusing with the egg cell to form zygote. That's why it is called syngamy. And the another one is called a triple fusion or endosperm formation, where the second male gamete will be fusing with the central nuclei to form the PEN, that is primary endosperm nucleus. That's why it is called a triple fusion. So three nuclei are involved in the fusion. You can see here one of the male gamete from the pollen tube is joining with the 2n number, forming 3n number, triple, 3 times. So that's why it's called a triple fusion. So these will be discussed in detail in the classes. Okay. So please remember the diagram based questions for board examination, type of pollination, self-pollination and cross-pollination, the difference, and also the double fertilization events, how it happens, and the terminologies associated with endosperm formation, syngamy, fusion of gametes, and the double fertilization. Okay. Good morning friends, welcome to today's class. Let's start the day's class with the quote. The intention behind the act is more important than the act itself. Today we will be discussing the post-fertilization events, the embryogenesis and seed formation with labeled diagrams. After fertilization, the degenerating synergids and the zygote and the primary endosperm cells and the primary endosperm nucleus and the degenerating antipodals you can identify in the post-fertilization cell, the embryo sac. Gradually the zygote develop into a globular embryo, then coming to heart-shaped embryo with suspensors suspending the radicle and cotyledons that form the mature embryo. Okay. Thank you for watching. Have a nice day.